Hi everyone, welcome back. I am very excited for today's lecture because it covers one of my favorite topics in science, which is transcription and translation. Now in chapter 7, we're going to explore the DNA structure and we're going to take a look how we can take certain sections of the DNA called genes and allow them to be converted into RNA and then into proteins. So without further ado, let's get started with chapter 7 entitled DNA Structure and Gene Function. Now, here's a quick overview of DNA. We have mentioned it in previous chapters, but I want to make sure that we're all on the same page. So thus far, we've discussed the fact that DNA, which stands for deoxyribonucleic acid, is our genetic code. It is composed of a double helix, which means it has two sides, and holding the sides are going to be a combination of what we call the sugar phosphate backbone and the nucleotides. Now, if you wouldn't mind, go ahead and take a look at figure 7.5, and you're going to see that the helix will kind of twirl itself around, almost like a spiral staircase or a set of pearls. And when you look at the spiral, you're going to notice that it has the backbone, which are the outer edges of the DNA molecule. This is composed of a sugar phosphate. Now, the rungs or the little lines that hold together the backbones, so these little things, those are going to be called your nucleotides. And the nucleotides are going to be your adenine, represented by the letter A, your thymine, represented by the letter T, the G will be for guanine, and the C will be for cytosine. So those are your letters, your A, your T, your G, and your C. Since DNA is a double helix, that will mean that the two letters will have to interact with each other to hold on to the two sides of the molecule. And in order to form the most stable DNA molecule, we're going to form this thing called the law of complementary base pairing. And that basically tells us that A on one side of the DNA will be hydrogen bonded with a T on the other end of DNA. And if you have a G on one side of the DNA, that will hydrogen bond with a C on the other end of the DNA. Now, is it possible to create hydrogen bonds between an A and a G or a T and a C? Yes, but that would mean that your DNA molecule would not be stable, which would mean most likely it would fall apart before its intended use can be benefited from. So please keep in mind, DNA, a double helix, so two sides. It is composed of the nucleotides, which are going to be represented by the letters A, T, G, and C. And the law of complementary base pairing tells us that in order to create the most stable DNA, A will go with T and G will go with C. Now, I want to take a few seconds out and have a little bit of a history lesson because I want to give credit where credit is due and name some of the scientists that were instrumental in the discovery of the DNA structure. Now, our story is going to start in the 50s, and in the 50s, scientists had already identified DNA as our genetic information. We knew that it carried our genetic code through the use of the letters A, T, G, and C, adenine, thymine, guanine, and cytosine. But what scientists weren't sure of is exactly how the structure was linked together. So the fact that it had a double helix and that it was intertwined, giving the appearance of a spiral staircase. So that mystery was solved due to the help of Crick, Watson, Wilkins, and Franklin. And I've inserted their picture at the bottom of the slide. And the way the story goes is that all four scientists were working on this question as to what the structure of DNA was. However, they were working on it in two separate groups. You had the team of Watson and Crick and the team of Wilkins and Franklin. And as the story goes, one day Watson and Crick were having lunch with Wilkins, and as they were kind of discussing their research, Wilkins mentioned that his colleague, Rosalind Franklin, was taking pictures of DNA and creating these X-rays. Upon seeing the X-rays, Watson and Crick were able to figure out that in order for the picture to match the function of DNA, it would require a double helix. So they went back to their lab and using a cardboard model, they went ahead and represented the spiral turning of the double helix. They then wrote a paper that was published and highly revered, and it turns out that in the 60s, to be exact, 1962, their discovery was actually awarded the Nobel Prize. Now, here's where it gets a little bit murky, shall we say. When the Nobel Prize was handed out, it was given to three people. It was given to Crick, it was awarded to Watson, and the third person was Wilkins. So what happened to Miss Franklin? Well, unfortunately, Miss Franklin had passed away in the 60s due to cancer, most likely due to the high amounts of x-rays that she was exposing her body to. So in that moment, she did not receive all the credit that she was due, because after all, it was her picture that was so instrumental in allowing these men to come up with the final form of DNA. So I always like to tell the story because Miss Franklin didn't get to share the Nobel Prize, but we do know her name now in history, and we know that she was instrumental in the discovery of the DNA structure. Now let's go back and take another look at our complementary base pairing that I mentioned to you on our first slide. Now here you can see the names completely spelled out of the nucleotides: adenine, guanine, cytosine, and thymine. And if you look at the illustration at the bottom, what you see is the interaction of the G with the C and the T with the A to create our stable bonds. The bonds are represented by the dotted line that you see right over here in the images. These dotted lines are going to represent our hydrogen bonds. Now, one thing I also want you to notice is that for each of the bonds that you create, one of the nucleotides is labeled as a purine and one as a pyridine. Now, a purine means that when you look at the chemical structure, it will have a double ring appearance. So it will have two rings based on its chemical structure. And I'm going to go ahead and number the rings 1 and 2. And as you can see, that will cover the adenine, the A, and the guanine. The pyridine indicate that it only has one single ring, and that will fall under the nucleotides for the cytosine and the thymine. So this picture then allows us to not only see if it has a double ring or a single ring structure when it comes to the nucleotide, but it also highlights the fact that in order to create a stable bond, the best thing to do is to mix a purine with a pyridine. So an A and a T and a G with a C. Keep in mind that these structures are being held together by hydrogen bonds. The other thing to mention about the DNA molecule is that when you look at its backbone, indicated here by the blue ribbon, that sugar phosphate backbone that I'd mentioned to you before, it will run in an antiparallel format. What that means is that when you look at the overall display of the deoxyribose, the sugar, we see that we have carbons in some of its corners. Now, the carbons that we're interested in are going to be what we like to call the 3' carbon and the 5' carbon. And they're indicated by the 3 with the prime signal on top, or the 5, once again, with the little prime symbol on top. Now, when I say that it runs anti-parallel, that means that if you look at your DNA model, and because of its two sides, it's going to have two backbones. If one backbone starts at 5, it will end at 3. And this all has to do with how the sugar is oriented. So the 5' will be pointing upwards, and then the 3' will be pointing downwards. The other side of the DNA molecule will run anti-parallel. So it will run the opposite way. So if this one started with 5 and ended with 3, its opposite ends will start with 3 and it will end with 5. Once again, this just indicates the orientation of the 3 and or the 5' carbon that we are looking at in our DNA molecule. Now, the reason I like to bring this up is because if you keep this in mind and you mix it with the law of complementary base pairing, it allows you to correctly predict what both sides of a DNA molecule will look like, assuming that you know what one strand is. So for instance, let's say we're looking at an example and I say to you, hey, when you look at your DNA molecule, one side of it, starting with the 3', has the nucleotides A, T, G, and C. So I'm just telling you the order of the particular nucleotide, so very similar to my image. You see them here on display. And depending on the order, that's how we're able to follow the blueprint, because that's how your cells are able to see what type of amino acid or proteins need to be produced. So if you know what's happening on one side, then I can then say to you, if this is one part of your DNA, what is the other side, right? Because it is a double helix. Now, in order to figure out what the other side is going to look like, we're going to have to do two things. The first thing is we're going to go and take a look at our carbons, and we're going to keep in mind that they're antiparallel. So if one side starts with 3 and ends with 5, that would mean that the other side will do the opposite. So we're going to start with a 5 and we're going to end with a 3. That's going to indicate the backbone running antiparallel. The next thing we're going to keep in mind is that the nucleotides, the letters, are going to follow their complementary base pairing. So if I have an A on one side, I am going to pair it with a T on the other side. So my A will pair with a T. My T will pair with an A. And then we're going to do the same thing for the G, but this one, of course, will pair with a C. And the C will pair with a G. Now, my little lines that I'm drawing right here, those are my little hydrogen bonds. So I'm able to see what one side of the DNA looks like by by knowing what the other side looks like. And even on your image, you can see it. Let me just kind of erase this right here for you so we can get rid of some of the markings. Even on the DNA molecule that's on display for us, we can see that the nucleotides are in the middle, and we could see that if there's a C on one end, the other side will have a G. If there's a T on one end, the other side will have an A. And once again, it doesn't matter if it's T and A or A and T, C or G or C. It doesn't matter which letter comes first, as long as you keep in mind that the law of complementary base pairing tells us that A and T go together and G and C go together. And as for the backbone, if one side starts with 5 and ends with 3, the opposite end will run antiparallel, so it will start with 3 and it will end with 5. And as you can imagine, our cells have a massive amount of DNA, and all this DNA needs to be condensed very tightly, and eventually it will be condensed all the way to what we like to call chromosomes, which are the discrete packages of your DNA within each of the cells. Now, humans, in case you were wondering, have 46 chromosomes, or we like to say 23 pairs, and that's because you inherit one set from mom and one set from dad. Your genome is the term that we use to describe all of your genetic material inside of a cell. And what we need to talk about now is how exactly can we use that DNA. Now, take a look at the picture, and what we're seeing is a simple bacteria cell, and all this little blue scribbles and ribbons that you see above it, that is all the DNA that's inside of that one cell. And in case you were wondering, bacteria only has one chromosome. So we have 46 chromosomes, so you can only imagine how much DNA that is in every single one of our cells. Now, part of what the DNA will do is that certain sections of the DNA will allow the cell to make RNA, and based on the RNA, we'll be able to produce proteins. And we can use proteins in a wide variety of things, anywhere from your enzymes to your hormones to building organ systems and your muscles. So it's very important. Now, not all of your DNA can be used, only certain sections. And what we're going to do now is we're going to take a look at that process of going from DNA to RNA to proteins. And in order to discuss that, we're going to have to be familiar with the terms transcription and translation. Transcription is going to focus on our first step of taking the DNA and converting it into RNA. And then translation is going to be the second step, which is now that you have RNA, how do we turn that into a protein? Now, not all of your DNA is able to be copied into an RNA, so oftentimes you'll see the mention of this term right here, a gene. And a gene is a particular section of your DNA that's able to do transcription. So the genes will allow us to copy our DNA into RNA. That process is called transcription. And then translation is when we take that RNA and we make it and we use it to make proteins. Humans have around 23,000 genes, and those genes are being held together in our 46 chromosomes. And using the information in those genes, we'll be able to make hundreds of thousands of different proteins for our body to utilize. Now, as for the location of where this takes place, you might recall from chapter 3 that DNA sits in our nucleus and it cannot leave. So the transcription step, the movement of DNA to RNA, will take place in the nucleus. And then the RNA will travel into the cytoplasm where it will then complete the second step, which is the movement from RNA to protein or translation. I like how the book describes it as thinking about making a dish or cooking. You know, let's say you are trying to cook your the brownies that your grandmother made for you. I'm just giving an example. Chances are that she has a recipe in a beautiful cookbook, and you don't want to take the cookbook with you when you go to the grocery store. So oftentimes what we'll do is we'll take a picture of the page. So your cookbook represents your DNA. That's our original source of our recipe. That's our genetic code. And when we take a picture of the particular page that we need, then what we're doing is we're making an RNA copy. And that RNA copy can then freely travel with you. And as you go through the grocery store, you'll be able to pick up your individual ingredients. In that case, the ingredients are going to represent our amino acids because as we discussed in chapter 2, when you put your amino acids together, you are able to build proteins. So DNA to RNA to protein, transcription and translation. There are a few differences between RNA and DNA. Obviously, we already know one of the differences, which is that DNA has to stay inside of the nucleus, but RNA can go ahead and leave. RNA also structurally is a different molecule. And there's this beautiful table that's represented on your PowerPoint that will highlight some of the changes. One of them is the fact that when you look at the name, notice that it starts with an R, not a D, and that's because the sugar is slightly different. The sugar for RNA is ribose, not deoxyribose. So that is why RNA stands for ribose nucleic acid. Another difference is that RNA, when it comes to the bases, it will have a U instead of a T. The adenine, the guanine, and the thymine will always be present regardless of what molecule you're looking at. But thymine, the T, is exclusive to DNA, and uracil, the U, is exclusive to RNA. So that means that when we're doing our complementary base pairing, we have to keep that in mind. Whenever we're binding DNA to DNA, the rule of A to T and G to C will flow. But if you're pairing DNA with RNA, you want to keep in mind that if you have an A, you can't bind it with a T because there's no T in RNA. So instead, you're going to bind it with a U. The G and the C will stay together regardless. They'll stay the same because both DNA and RNA have it. And if you have a T in DNA, you can still bind it with an A in RNA. So really the only base pair that's affected is the A in DNA, which will have to bind with a U in RNA. Below, you also see an analogy or an example of how RNA will be base pairing. So as you can see, G and C will stay the same, but in this case, the A and the U will interact because there is no T. The last thing I want to mention to you about RNA is its form and its function. The form of RNA is that it's single-stranded, not double-stranded. So DNA has the two sides, RNA only has one side. And the function of it, of course, will also be different. DNA will have your genetic code and will be able to produce RNA, and the RNA will carry the copy of that DNA and will allow us to produce proteins within the cell. There are three types of RNAs that are going to come into play when we start talking about translation, the second step of taking RNA and making into proteins. So I want to go ahead and start introducing them so that later on when we see them interact, it will make sense what each of their role is. The first type of RNA is called messenger RNA, and messenger RNA will carry the information. This is the exact copy of what the DNA has of its gene, so we will be able to use that copy to figure out what protein we need to produce. Now, ribosomal RNA will be able to read the message, and with the help of ribosomes, we'll figure out what protein needs to be produced. This will be the location of the protein synthesis, because in a little bit I'll show you an image of how the RNA molecule can clamp onto the messenger RNA and read its directions to figure out what amino acids need to be gathered. Speaking of amino acids, the one that will collect and bring them over will be the third type of RNA, and that will be our transfer RNA. Transfer RNA will scavenge the cytoplasm and look for the particular amino acids to bring over based on the directions of the mRNA. Once these amino acids are brought over, they will be strung together and put together with peptide bonds to allow for the polypeptide or the protein to be formed that is required based on the genetic information that is being transcribed and translated. In order to do our first step, transcription, so take our DNA and convert it into RNA, there are going to be 3 steps that we have to complete. Those 3 steps will all take place in the nucleus, and they are initiation, elongation, and termination. Initiation will consider the fact that your DNA, which is a double helix, will need to be opened up almost like a zipper so that the 2 sides can display their nucleotides. They can display their letters, their A, their T, their G, and their C. In initiation, we see that the DNA is unzipped, and we're going to utilize a particular enzyme called the RNA polymerase. See how it ends with ASE, so it tells us it's an enzyme. This RNA polymerase will go ahead and it will bind to the DNA, so it will assume the position. Once it's in place, we will move on to the second step, which is called elongation. And in elongation, what's going to happen is that the enzyme will start to move along the DNA strand from the 3 to the 5 prime, and it will read the DNA molecules. And if it comes across the nucleotides, it's going to start matching it with its complementary strand of RNA. So if the RNA polymerase comes across an A, it's going to match it with a U, because remember, there's no T in RNA. If it comes across a T, it's going to match it with an A C with G, and a G with C. And in elongation, the RNA polymerase just keeps reading the nucleotides and matching it up to its complementary base pair. When the RNA polymerase comes to the end of its track, when it's all done, it's going to transition to the third step, which is termination. Termination means that we have reached the end of our sequence, so the RNA polymerase can fall back off. We no longer need it. The DNA will curl back up into its double helix, and the brand new RNA strand is now ready to move from the nucleus into the cytoplasm in order to transition to our next step, which will be translation. Your body, or I should say your cells, will have to do these 3 steps regardless if they're doing mRNA, tRNA, or rRNA. Now, here are a few illustrations to highlight the steps of translation. Once again, our goal is to go from DNA to RNA, and this is all happening within the nucleus of the cell. So in our first step in initiation, we see that the DNA will unzip itself, and then the RNA polymerase enzyme will attach. This is the one that's going to be doing all the work. So initiation is about prepping it. Get it to attach and get the DNA to unzip. Then in elongation, we're going to see that the RNA polymerase starts to move. And as it's moving, it's reading the DNA. You could see it here in the zoomed in example. Blue is DNA, green is RNA. And you could see that as the RNA polymerase is coming across the nucleotides, it's matching it to the complementary base. So C with G, A with U, G with C, and it keeps going. And as long as the RNA polymerase is moving and reading the DNA and making RNA, it is in the elongation step of transcription. Finally, we'll get to the end, and when we get to the end, that step is going to be called termination. Termination means that the DNA will reach its terminator sequence, it will reach the end, so the RNA polymerase is removed. Bye, see you later. The DNA will coil back up to its original structure, and the RNA molecule is ready to go. So remember, the RNA molecule is important because not only does it carry the copy of the DNA sequence, it will also be able to move into the cytoplasm so that we can utilize it to make our proteins. I also want to mention that in order to increase the success rate of the RNA, your cells will go ahead and try to increase the stability of the molecule. And this will primarily be for the mRNA, the messenger RNA. And the way we're going to increase the stability is by doing a few things. The first thing that we can do is we can go over to its 5' end and we can add what they call a cap. And a cap simply means that we're going to fold it over and create a little cap right here, this little folding over strand. That happens on the 5' end. The 3' end, the other end of the mRNA, is going to get what they call a poly A tail. And that means that we're going to take a bunch of adenines and we're going to string them together. Hundreds of them will just get added into a tail format. And it turns out the longer the tail, the more stable the mRNA is going to be. Please keep in mind that the 5' will get the cap and the 3' will keep the poly A tail. It is very specific as to what location will go where. So 5 for the cap and 3 for the poly A. The other thing that the RNA will do is it will do splicing. Splicing will help the RNA get rid of the items that it doesn't need and hold on to the sequences that are important. So in your book, it discusses that you have within your DNA and your RNA, you have sequences that are considered introns and you are sequences that are considered exons. The important ones are the exons. They're the ones that are expressing the mRNA, and they are the ones that will exit the nucleus. So see how the EX will help us remember the important one? The introns, for all purposes, can be considered junk. We no longer need them at a certain point, so why carry them on? Let's splice them out. Let's cut them out. Let me show you this in an illustration. So here is my DNA molecule. This is all happening within the nucleus, and in transcription, we went ahead and we created our mRNA. Now, in order to increase the stability of the mRNA, notice how they went ahead and they added the cap on the 5' and they added the poly A tail on the 3'. The next thing that's going to happen is that it's going to get a little bit shorter. And the reason it's getting shorter is because we're going to splice. And when we splice, we're going to get rid of the introns because we don't need those anymore. And we're only going to keep the exons. This will then create the most stable version of the mRNA, which will then allow it to be transported into the cytoplasm so we can do our translation step. And a fun fact is that the exons don't always have to go back in the same order. So as you can see in our example, the exons are A, B, C. They could have also been put together in a different way. So maybe A, C, B, C, B, A, B, C, A. And it turns out that by creating different combinations of exons, we're able to create different proteins at the end of doing translation. This is one of the reasons why we have only 23,000 proteins, 23,000 genes, but we're able to create hundreds of thousands of proteins by just altering the way that the exons are put together towards the end. All right, so now that we've done transcription, let's talk about translation. Translation is going to be all about reading the mRNA and producing the protein that the cell requires. It's going to take place in the cytoplasm. Now, in order to explain translation, I have to tell you that we're not going to read the mRNA in one full strand. Instead, what we're going to do is we're going to break it down into what they call codons. A codon means it consists of 3 bases. So your mRNA, we're going to look at it and we're going to read it 3 letters at a time. So let's take a look at the image that's at the bottom of our screen. And we could see right here that we did our DNA. Here's our template in blue. And after transcription, you have your mRNA. So you could see the DNA was red, A with T, C with G, A with U, and it was matched with a corresponding RNA. Now that the mRNA is nice and stable, what we're going to do in translation is we're going to read it to figure out the proteins we need to produce. But we can't read it in one sitting. Instead, we're going to read it in blocks of 3. And these blocks of 3 are called codons. And the reason a codon is so important is because your body has figured out a way to have specific codons linked to specific amino acids. So when we do translations, we're going to read the 3 letters at a time. We're going to read the codons, and the codon will convey to the tRNA which amino acid it needs to find and bring over. And as we collect the amino acids, we will start to form our polypeptide, which will give us the protein that we're doing all this work for. Here's an example of what we call our codon table. This is your genetic code on full display. And what this table is showing us is all the different ways that the 3 letters, or I should say 3 letters of your nucleotides can combine to create a codon, and what each codon can represent based on the amino acid it's looking for. So for instance, let's go ahead, let's take a look at this little box right here. If your body needs the amino acid proline, these are the codons that will give it proline. And as you can see, it has lots of options. There's some more above it, some more below it. And notice how all the combinations involve only 3 letters, 3 nucleotides. So these are your codons. Now, the best way to read this table is to follow the directions, which basically tell us that we want to start on the left-hand side and find the first letter of our codon. Then we're going to move to the side for our second letter, and then we're going to look within the box for our third letter. So let's say, for instance, on your exam, I say to you, what amino acid will you find if you look for the codon G, G, C? So what amino acid will represent the codon GGC? So I will provide you with the codon table on your exam. And when you take your exam, you're going to be able to see that we're going to start on the left-hand side, find your first letter, the G. Then we're going to go across the way for the second letter. So staying in here, we're going to find our second letter, which is another G, which will bring us over to this box right here. And within this box, we can look for our third letter, which is the C. Here it is, GGC, which will give us glycine. Now, a few things. One is that the third letter of the codon is often what we like to call in a wobble position. It doesn't really matter what letter you put there. Chances are you're still going to get the same amino acid. This is really like a safe mechanism for mutations. If the mutation happens at the third letter of the codon, most likely the mutation will be what we call a silent mutation. It won't have an effect on the overall amino acid that need to be brought over. And in our example, the glycine is perfect because it doesn't matter if at the end of the day the third letter was a U, a C, an A, or a G, it would still produce the same amino acids for us. It doesn't always work that way, but your body has it set up that way anyways. The other thing I want to mention to you is when you look at your table, notice how you have a start and you have a few stop codons. These are going to come into play when we talk about the steps of translation. And on your end, I don't want you to worry about memorizing the codons because we can reference the table. The only one that I want you to memorize is what the start codon is. The start codon will always be AUG. And the reason I want you to remember that is because it's a universal codon. Every single organism that you'll ever come across will always have the same start codon, AUG. Personally, I remember it as the month of August. So when you get your codon table from me on your exam, it's going to look just like that. So you can reference it for the questions. But what I will do is I'm going to remove the start codon mention so that one of your questions can be, what is the start codon? And you will look for an answer that's going to say AUG. Now, I said to you before, the tRNA, the transfer RNA, is the one that's in charge of going to get the amino acids. So it's the one that's going to be able to go and get the proline, the valine, any amino acid that's required. When the tRNA brings over the amino acid, we have to make sure that it puts it in the proper spot. So in order to do that, we're going to see that when we take a look at the structure of the tRNA, it's going to allow us to have what we call an anticodon section. The anticodon is the complementary match to the codon of the mRNA. So for instance, if my codon is AUG, yeah, you're right, that is the start codon, then the tRNA anticodon would be UAC. So it's going to be a complementary match to each other. And the reason you need that anticodon is if you look at your picture right here, this is what your tRNA looks like. You'll notice that on one side of it, it will have the amino acid that it's carrying, and on the other side, it will have the anticodon. So when the tRNA brings over the amino acid, it wants to make sure it's put in the proper location, so it will temporarily bind with the codon on the mRNA, and it will be able to bind because it's a complementary match. Once it makes that covalent bond, it's very quick and it confirms that it's in the right spot. It's a perfect match to the codon and the anticodon. It will then release the amino acid so that we can start building or continue building our polypeptide chain. So how does this all fit together? We know that we have our translation step. We know that we have to take our codons and we have to get our amino acids and match it with the anticodons. But how does this all fit together with the mRNA, the tRNA, and the rRNA? Well, let's go ahead. Let's take a look at the 3 steps. The name of the steps are going to sound familiar: initiation, elongation, and termination. They get the same names as transcription, but obviously the events will be different because our goal is now to take the RNA and make it into a protein. And this, of course, is happening all in the cytoplasm of the cell. So let's take a look at the events that happen in each of these. Let's start off with initiation. In initiation, what we see happening is that we have our mRNA strand. And the mRNA, remember, we're going to read as codons, so 3 sections at a time, 3 letters at a time. And in initiation, what's going to happen is that our rRNA, our ribosomal RNA, is going to go ahead and attach its small subunit to the mRNA. So here's my rRNA. It's going to attach it to the mRNA, and it's going to start looking for the start codon. The start codon is not always at the beginning of the strand, so it's important that the rRNA finds its proper start codon. So in our illustration, you can see that it locates the AUG. That's our start codon. And at the end of initiation, once the AUG is located, we see that the large part of the rRNA is brought over to attach to the small part of rRNA. rRNA is composed of 2 pieces, a small subunit and a large subunit. The small subunit will find the AUG, and then recruit over the large subunit. Once these two pieces are together and the AUG has been identified, then initiation is over and we go to our next step, which is elongation. In elongation, we see that the majority of the work is done. So once again, we have our mRNA sequence here in green. We have our rRNA, the small subunit and the large subunit. And the rRNA is now going to start reading the codons. And as it's reading the codons, the tRNA is bringing over the amino acids that are associated with it. And keep in mind that when the tRNA comes, it has to make sure to put it in the right spot. So it's going to match its anticodon with the codon. And in elongation, we just continuously keep reading these codons. So you can see on my picture, the rRNA is moving along, the tRNA is depositing the amino acids, and here in purple, that's representing our amino acids, and we keep growing the polypeptide chain. And it will keep going, reading codons over and over again. Eventually, we're going to get to our last step, which is termination. In termination, we reach our stop codon, and the stop codon will then tell the rRNA that we are done. So the rRNA will fall off. It will also release the protein, the polypeptide, and we are now at the end of our translation, which is that we were able to produce a beautiful strand of amino acids, and we're able to create the protein that our cells were after. So once again, in order to do translation, we start with our mRNA and we do initiation, which means we find our AUG, our start codon, and properly attach our rRNA. Then we do elongation, which is that the rRNA is reading the codons of the mRNA, and the tRNA is bringing over the amino acids and matching its anticodon to the codon so it knows its proper location. And last but not least is what's illustrated right here, which is termination, which is we reach our stop codon and we have a brand new piece of protein for the cell to utilize. Now, one of the things that I would love for us to do is to do an example of transcription and translation. So when we meet for class, I'm going to talk to you about your assignment, which is that I will be providing you with a piece of DNA, and you will go ahead and on paper complete transcription and translation. So I look forward to talking to you about that assignment and for us to actually be able to see how we can create RNA from DNA and how we then read the codons and use the codon table to figure out which amino acids are needed for our protein. All right, per usual, if you have any questions, comments, or concerns, please do not hesitate to reach out and we shall talk again soon. Bye, everyone.